giving an international lecture with 500 attendants. Yes, 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 yes.
Okay, so we are streaming on YouTube now.
All right.
Yeah, we still have a few minutes.
Yeah, I have 4 minutes left. Nancy is happily grinding some coffee in the background.
Just once.
We're streaming on YouTube now.
All right. Yeah, 4 minutes left.
Hey, Martin, I just David Nesbitt here. Just want to know
Hey, hello.
how's chairmanship being? Are you still
It
chair?
was good.
Good.
So actually, I stepped down from department head on June 1st.
Bravo. Good for you.
Perfect time.
Who's taking over now?
Kathy Murphy.
Oh, that's right. That's right. Kathy said, yeah, that's right. Well, she'll be good too.
Kathy. Oh, she's very, very good. She'll be better than I ever was.
Well, we'll say she will aspire to be as good.
Yeah, absolutely. Absolutely.
Good.
Yeah, I'm enjoying my copious free time writing up papers.
Right, exactly. Is Illinois, you guys, I know that Kathy was telling me that you guys are opening up your, in a first stage of opening.
Yes, so we allowed researchers back into labs starting June 3rd with strict guidelines on distancing, face masks, and so forth. And we haven't had any incidents in the last month. So far it's worked pretty well. Is
Illinois one of the few states which is not increasing its number of COVID?
Illinois has a bit of an increase, but one of the lowest ones. So people here stuck pretty well to the guidelines, and our governor was one of the very first after California to, take measures. So we're actually in pretty good shape.
Well, good. I should let Gil sort of monitor this since you're supposed to be speaking, I guess, now.
So. We'll let Ben take over with his introduction.
No worries, Dave. We will start in a minute.
Yeah, we'll just wait another minute or so that people can join, and then we'll get going.
Well, let me just say, are your students, are your undergraduates responsible enough not to have parties and get-togethers?
Well, so we don't have them on campus, right? They were all sent home during spring break. So the only undergraduates that are really around right now are ones that were doing research before the crisis started, that also don't need any training from graduate students. In the fall, they are planning on doing mixed online and in-person instruction. So it's going to be quite adventurous. I'm interested in seeing how that's going to work.
Yeah. Yeah, well, Boulder, of course, has a large student component in the town itself. And
Yes.
it's hard to get the younger people to appreciate the seriousness of social distancing.
It is hard. It's hard for everyone.
Okay, so maybe we should get going. So I would like to welcome all of you. Great to see so many of you back. Familiar faces, familiar names. And before we start, maybe a few organizational things. I would like to ask all of you to keep your microphones muted during the talk. There will be plenty of opportunity to ask questions at the end. Please use either the chat window and just say that you have a question or put your question there, or raise your digital hand and then we will call you in the end. I would also like to mention our YouTube channel, which you can find on our website, which is shown here. And I would also like to mention already our next speaker in 2 weeks, which will be Jane Dyson. And so now I would like to extend a particularly warm welcome to Martin Gruebele, who is our lecturer today. And as most of you know, probably, Martin is one of the most versatile and innovative scientists around. And his interests are correspondingly broad, from the imaging of electronically excited quantum dots to glassy dynamics of materials, to advanced laser spectroscopy and microscopy. And that's what most of you probably know him for, because that's a range of techniques that he's applied extremely successfully to protein folding and dynamics, but also to questions like vibrational energy flow. And I've known this all for a long time, but then I was still very surprised a few years ago when I heard Martin give a talk. Then he suddenly started talking about the swimming motion of zebrafish and how they used singular value decomposition to decompose it into eigenshapes, and how they could show that 3 eigenshapes are enough to capture more than 96% of zebrafish motion. And so he's always good for a surprise. And this kind of work also illustrates another key characteristic of his work, which is the combination of fantastic experiments with very clever analysis, theory, and computation, and just very deep thinking in general. But not only in general, but also within the field of protein folding and dynamics, he's been extremely versatile in his work, which has ranged from fundamental questions that he addressed, for instance, using laser spectroscopy, such as the folding speed limit and the comparison of experiments and computer simulations. Also method development, like ultra-fast pressure jump or T-jump in live cells. And this is something that, a topic that brings us to one of the topics he will also be talking about today. That's the questions of crowding and the role of cellular factors for protein folding and stability. And so his versatility already shows up directly in the title of his talk, which will be Protein Folding and Association Dynamics, from in silico to in vitro to in vivo. So with that, thank you very much again, Martin, for joining us and being our lecturer today. And with that, I will stop sharing my screen, and the screen is yours.
Martin, are you online?
Yeah, I'm here. Is my screen being shared?
Not yet.
No?
Yet. Okay, so let me actually maybe turn that back on. There we go.
Okay.
it says I'm screen sharing. Can everybody see the title of the presentation?
Yes, there it is.
Ben, yeah, I'll count on Ben for my feedback. Also, Ben, if it turns out that my internet should drop out temporarily or something like that, just wave your hand or make some signal and I can repeat my sentence just in case.
Okay.
Since I am, like all good Illinoisians, I am talking from home, where all people who don't need to be in a lab, like research advisors, are supposed to work from. So hello, everyone. And I really would like to thank Gilad and Hagen and Ben for organizing this series of seminars, which is giving us interesting stuff to hear from a variety of people over the last couple of months. I've enjoyed them a lot, especially because I had to cancel every single one of my summer talks and flights and everything. So no whizzing around in Switzerland or Israel or anywhere, instead sitting at home. So I'm going to talk to you today about protein folding and association dynamics from in silico to in cell to in vivo. I cut out a little part. Ben mentioned in vitro. There was just no time really to do this in like 45 or 50 minutes. And so I eliminated that part from the talk. So let me launch right into it. My research group is visible there in the courtyard in front of our chemistry and biology buildings. And the work I'm going to talk about in particular today was done by some recent graduates and one person who's still here, Caitlin Davis, who is now at Yale University on the faculty, Rupei Feng and Drishti Gwyn, who are both in the Bay Area doing data analytics. Meredith Rickard, who is actually up here in the background, smiling at us, right there. Shahar Sukenik, who is at UC Merced now, and Yi Zhang, who is working to be a rich banker somewhere on the East Coast. So a wide variety of kinds of places and things that people like to do with their PhDs in chemistry or biophysics. And some of the work is in collaboration with Professor Taras Pogorilov, also in the chemistry department here. So let me launch right into it. Here's my very short in vitro slide. One of the main things that we've learned from in vitro protein folding and protein interactions is that proteins have shallow energy landscapes. So the saddle points or the free energy barriers are only a few kT higher than the local minima. And the free energies of local minima, like the folded or unfolded state or the bound or unbound state, are also not really very different from one another. A few kT, a few tens of kilojoules per mole on that order in many cases. And so for those kinds of chemical reactions with relatively low barriers and relatively small free energy differences, you might expect that they could be sensitive to the environment, right? Because it doesn't take much of an effect of salt screening or changing the temperature or changing the pressure or some other parameter to actually shift these free energies around. And so this could lead to modulation in the cell of protein landscapes. And we can conveniently divide the contributions into two different parts, repulsive and attractive. So by repulsive, what I'm really referring to is crowding or excluded volume. And the idea there is basically that volume reduction leads to a reduction of configurational entropy of macromolecules. And so, for instance, that might favor a compact folded state over a non-compact unfolded state. Again, it's a small effect, inside a cell. Even if you take the most voluminous, water-containing cells versus the driest, we're talking about maybe a 50% volume reduction if you put cells into a strong osmotic solution and drive most of the water out of the cell. So again, it's a few kT effect. But basically, we're talking about the repulsive force, which in the simplest cases, people actually like to approximate it kind of by repulsive spheres, and that protein there in the middle, or that molecule in the middle, kind of looks spherical enough. And then on the other hand, we have attractive forces, of course, and I'll refer to that as sticking when it's non-functional. But of course, often things are supposed to stick to one another. Proteins are supposed to stick to themselves when they fold. They are supposed to stick to other proteins or RNA molecules or other molecules when they do signaling or when they bind for various reasons. And then I like to use the term plenary structure as a level of organization above quaternary structure where complexes form, but they're not as tightly bound and they're not as long-lived as what we typically think of when we talk about a quaternary complex. So these are transient weak attractions, and even like charges can attract. So in this little example picture I have here, this is some in vitro research. I guess there is my in vitro slide here that we did about 10 years ago, where we basically took small angle X-ray scattering spectra of a large number of concentrations of this protein SH3, a small beta sheet protein. And this particular mutant we're using has a charge of -4 elementary charges per protein. And so you would expect some kind of screen repulsion initially, and that's what we see. When we do these SAX experiments and we analyze them as a function of concentration, we developed a method that we can directly extract numerical potentials of interaction out of the data. And what we obtained in this particular case by two rather different methods are similar looking potentials, where yes, indeed, there is a repulsion initially when the two proteins are pretty far apart, but then finally we dump into a minimum, again, a few KT, same kind of free energy scale that I had for the crowding on the other side. And what's happening here is that even though, of course, like charges normally should repel one another, eventually you get them close enough proteins, so the water layer between the two of them, it becomes confined. And at that point, you might actually lower the free energy by just squeezing the water out, allowing it to move around more freely in the solvent and just sticking these two proteins together, even though the charges repel one another. So this is a like charges can attract case. But again, the point here being that the stickiness, this potential well, is on the order of a few KT. So quinary structure, therefore, might be particularly susceptible to environmental modulation. And one thing I think that's interesting to point out here is that the surface of a protein really can only encode for so many interactions. There's only so many bits of information available on the surface. There's a finite number of amino acid, tends to 100 or so on a typical protein surface. And so you kind of have to make a choice. You could have some interactions that use up a fairly large fraction of that surface and become highly specific, high affinity, right? Or you could have a protein that interacts with 10 other proteins, different parts of the surface, in which case those 10 interactions are not likely to be as strong. So that's, I'm encoding that in this sort of information entropy here, where basically the ratio of the surface area of a protein to the surface area of an amino acid that's exposed at the surface of a protein, if we assume that's kind of a potential recognition site. And then the fact that there's 4 general types of interactions, positive, negative charge, polar, non-polar, so that gives me that factor of 4 here. So there's a certain amount of information that's basically going to be available. And the paradigm used to be, and it's still true for many proteins, that one protein, one complex, you have a particular function, you have nanomolar KDs. And in cases like this, the interaction landscape is probably not going to be very strongly perturbed by the surroundings. But on the other hand, you can also have a protein that can have many functions and interact with many different things. HSP70, or actually I should rather say HSC70, which is the latently expressed one in the cell, is a good example. This chaperone plays a role in clathrin-mediated endocytosis, in ratcheting polypeptides through membranes, and of course, as a holdase, as chaperoning or holding other proteins. And so those kinds of proteins that have a lot of different functions often have KDs that are not as small, so micromolar, they don't bind as strongly. And the interaction landscape might actually be significantly perturbed, so that maybe it's not really realistic to try to use typical in vitro assays anymore, because they might actually perturb the binding or the folding or the thing that you're looking at. And so then you have to really look at in-cell assays or in vivo assays to see what's going on. And so I'm interested in the cases that are more on this end of the spectrum over here. So I'm going to give 6 examples of this, each just 2 slides. And going in great detail through a single topic, because this is an overview lecture. I know there are people here from many different backgrounds with many different interests, and so I thought this might be the best way to go. Some of the things I'm going to talk about, I think the oldest one was published a couple of years ago. Most of them are very recent publications or not out yet. So it's sort of a mix of recent and hopefully soon to be seen in the literature, the referees being kind subjects that I'm going to talk about. So number one, stickiness of the cytoplasm in current force fields and folding. So this is going to be some simulation work that Meredith Rickard and Taras Pogorilov did. And number two, I'm going to talk about quinary interactions between two enzymes, the good sticking. And number three, interference with lysosomal subunit assembly as an example of bad sticking. Sometimes things should interact with one another, and there's thousands of proteins and RNAs and lipids and all kinds of other things inside a cell. Sometimes you can't avoid sticking to things that you maybe don't really want to stick to. And then number four, preemptive chaperoning in cell by HSP70 and on time by HSC70. So I'm going to contrast two different weak binding or chaperoning mechanisms. Number five, cytoskeletal modulation of target proteins inside the cell. So this is more of a question of, if I change like the overall makeup of the cytoplasm by taking a cell and disrupting its cytoskeleton so it becomes more fluid inside, what kind of an effect, for instance, will that have on protein stability and dynamics? And then finally, another fairly recent paper, the number five isn't, I think literally might just be coming out today or tomorrow or something like that. Number six, folding in tissues. And this is basically asking the question how different tissues in an animal will actually host the same protein as far as protein stability is concerned. So let me launch in topic number one, and I've got them color-coded for you up here. Oops. So stickiness of the cytoplasm in current force fields and folding. And on the left-hand side, you see a snapshot of one of these simulations that we run on the Anton 2 computer at the Pittsburgh Supercomputing Center. And we've gotten interested in simulating a small chunk of cytoplasm. So we can't do a really big piece atomistically for long times, which was what it takes to look at folding. And so our little mini cytoplasm has 16 different types of proteins in it, 262 small solutes, 400 ions, about 50,000 water molecules, and it's in a distribution that corresponds reasonably as closely as one can with something this small to the E. coli cytoplasm. And we looked at the sticking dynamics of proteins and also at the folding of a small WW domain, that's a triple-stranded beta sheet protein, and the GTT mutant of it that we made a few years back is particularly fast folding. So it folds in a few microseconds. We've accumulated over 200 microseconds of simulations in this environment. The orange molecule here is the WW domain. You can see some ions and you can see some of the small metabolites dissolved in the cell, and then the other macromolecules in this sort of light blue or purple. And the simulations were done in a variety of different versions of the CHARM force field 22 star, 36M, and 36M CU-fix, where CU-fix is an attempt to correct, from my colleague Alec Aximentev in physics, to correct some of the Lennard-Jones type terms to make things actually less sticky, because the current generation of potentials might be a little bit too sticky. That's good when you're trying to fold a single protein in a water box, but it's not good if you're trying to simulate a cytoplasmic environment where there's actually lots of other things to stick to, and then you don't want to overdo it. What we found in the simulations, with that caveat, that there's probably a little too much sticking going on in the calculations, is that there is a power law distribution of sticking times between folded proteins that goes from close to unity when you're talking about just a survival time of a few nanoseconds for the sticking, down to about 1% of the contact events where sticking occurs for a microsecond or longer. But nonetheless, you get these encounters in the cell where things sort of come together and they stick to one another for a short while and then come apart again. There is a lot of emphasis these days in the literature on looking at the electrostatics of this, which is a very important contribution. We found in our simulation there's also significant contribution from hydrophobic patches on the surfaces of proteins that will stick to one another. Although, and the balance in our simulation came out to be about 50-50. Whether that's really true is not clear at the moment, simply because, again, the force fields are not quite good enough to really make that fine differentiation between, how much of it is going to be somewhat longer range and electrostatic and how much of it is going to be shorter range and hydrophobic. Let me show you some specific examples, though, what happened to the WW domain inside this mini cytoplasm. So when we do the simulation in vitro, so basically just a buffer solution on the computer, okay, what we observe for the protein typically is very large fluctuations in its radius of gyration. So it really expands and contracts and switches back and forth a lot. And then eventually it actually folds rather rapidly. In this case, it took about 18 microseconds for the protein to fold. And we're now at the native radius of gyration. So here there was really a folding event. What we see in our mini cytoplasm in contrast is there is some of these fluctuations, but actually less so. And instead, what the protein frequently does is it'll actually spend its time stuck to a surface in this cell model and fluctuating much less, and then coming free again for a while, and then being stuck again on the surface somewhere for a while, and then coming free again, and then it's being stuck again. You can see actually at the end here, it got stuck for a really long time, like 125 to 190 microseconds. This particular WW domain and that particular simulation among the three simulations we did, really got stuck. And when you actually look at what happened in this region here between 123 and 178 microseconds, so out here, is it ended up near two proteins, MDH and an elongation factor, EFTU, that we had in our simulation, and basically started making salt bridges. So the brown molecule here is the WW domain, and then here in purple and in white are the other two proteins, that are shown here on the right-hand side. And every time this diagram turns red is when a salt bridge forms, or every time it turns red here is when there is a strong hydrophobic interaction, let's say between phenylalanine on the WW domain, and in this case, another phenylalanine on the EFTU. And so basically, this is what happens. The molecule sometimes comes free and fluctuates around, and then very often gets stuck on the surface. And this is going to slow down the folding process. In fact, in this particular simulation, we never observed complete folding, even though this molecule takes only a few microseconds, and it's easy to observe nowadays on fast computers, complete folding and unfolding multiple times, in the simulation, just in vitro. We never observed it in our cytoplasm model. And we don't really think that the cytoplasm slows down the folding dynamics really that much in reality. There is probably some extra sticking in the calculation, but it shows that cytoplasmic sticking is going to have an effect on the folding kinetics of proteins. More dramatic things can happen. So here's an example where the WW domain has gotten into a misfolded state where it's formed helical structure, and it tends to do that actually more in our in-cell simulation than it does in the in vitro simulation. And this molecule, I think if I remember, is superoxide dismutase. And what happens is the WW domain diffuses in, makes contact with the other protein, and actually causes partial unfolding of this alpha helix that's shown here. And then it diffuses away again for a while. But then actually after about 58 microseconds, it comes back again, and it forms a chimeric structure where this alpha helix, or what used to be the alpha helix in this enzyme, now actually interacts and makes an anti-parallel beta strand structure with part of this misfolded WW domain. And then it eventually drifts away again, but after 106 microseconds in the simulation, actually comes back and forms that same beta sheet structure again. So this wasn't just a one-time hit. I mean, the molecule will diffuse away pretty far and come back and still make that interaction again. That leads us to believe that, this is a plausible interaction that could happen. And this kind of happening could actually, under stress in cells, be a mechanism that starts generating non-native like beta sheet structure that could eventually build up into oligomers and finally even worse things. So when you look at where these contacts form in the WW sequence, actually the highest propensity for forming this contact with our enzyme is this GTT, the mutation that we made to make it fold really fast. So this is a good example of just because something folds really fast in vitro is not necessarily good for it in vivo when it's actually being simulated in a more cell-like environment. So this little piece of sequence here, the folding, it could very well also be the reason why this WW domain here is doing more sticking to other proteins inside the cells. So anyway, with the caveat that current force fields do overdo the sticking somewhat, nonetheless, there's a real balance between sticking and crowding in the cytoplasm that can affect, in this example here, for instance, folding dynamics. So let me go to my second example, quinary interaction between two enzymes, the good sticking. The experiment that you're seeing on the upper right-hand corner here was done by Shahar Sukenik, now at UC Merced. And he basically took a cell and gave it in fluctuating concentration of lights. You can use, let's say, sodium ions or mannitol or others. And what happens is every time the osmotic pressure goes up, water is drawn out of the cell, and you see these black little holes appearing. These are places in the cell where the cytoskeleton is not protecting the cell from shrinking. So what's happening is water goes out and the cell shrinks down. And in these dark areas is where the cytoplasm has become really thin. So you don't see a lot of fluorescence from the protein in the cytoplasm. But this is completely reversible. You can see it appear and disappear as we go to lower and higher osmotic pressure. And the cell in a tube where we can just change volume. And at small volume, we get more association, right? Because it's a bimolecular or higher molecular reaction that's driven towards association at small volume. Well, it turns out you have to be a little bit careful with your design of the FRET probe for an experiment like this. For instance, when we used ACGFP1 as our green label on one of the two proteins that we're going to look at associating, that doesn't work very well. There's actually a FRET even of the label just by itself. But it turns out MEGFP in this particular cytoplasm with that particular binding partner here, there is no significant interaction. So that was a good label to use because we don't want to monitor the interaction of our two labels that we're using to look at the fluorescence. We want to monitor the interaction of the proteins that are binding to one another. So what happens in this experiment then is we have two proteins that are FRET labeled, one red and one green. And as they bind, as the cell shrinks, the red fluorescence increases and the green fluorescence decreases. And then when the cell expands again in the next cycle, the green fluorescence increases and the red fluorescence decreases again. And we can look at that switch. And so what we did is we looked at the interaction between two enzymes, 1,3-bisphosphoglycerate, that takes that makes this molecule bisphosphoglycerate and sends it on to phosphoglycerate kinase, which then abstracts a phosphate group on that to turn ADP into ATP. So these basically are part of the ATP generation cycle. And what we found in this experiment is, as one might expect, is that if you shrink the cell volume, the red fluorescence goes up and the green goes down. So we've labeled the one of these with a red label and the other one with a green label. So that means there is FRET and the two are interacting, the GAPDH and the PGK. And if I increase the cell volume, then the opposite happens. There's actually a very slight increase in the green and a slight decrease in the red as the association decreases. And so it turns out we can actually build a model that allows us to fit both an effective dissociation constant as well as the stoichiometry of how many GAPDHs and how many PGKs come together. And we found the best fit, which is shown as the dark area in this curve, corresponds to roughly alpha equals 2, beta equals 1. So a 2-to-1 ratio of this GAPDH complex versus PGK with a 14 micromolar affinity. So a relatively low affinity, nothing nanomolar here inside the cell. And so you might ask, well, if that, so why, first of all? And we think a good hand-waving explanation is that this kind of association between enzymes allows a faster diffusion of the substrate that's being made by the first one as the substrate of the second enzyme, right? Rather than the two floating around randomly inside the cell. But then you might ask, hey, why not tether the enzymes together? That's so good, stick them together permanently. And actually, there are plenty of cases like that inside cells, but not this particular pair. And one possible reason is that it turns out it was in the last couple of years figured out that PGK also acts as a kinase that can actually interact with a membrane protein. And this might actually act as a general health sensor for the cell. And so PGK actually has its main activity, which is making ATP, but it has other functions inside the cell. And so again, you remember I talked about the entropy of how much information is available on the surface of a protein, how much can you encode? So again, if a protein has multiple tasks, then maybe permanently linking it to one of those tasks is not the best thing to do. And so maybe it's not entirely surprising that PGK is not necessarily tethered to the GAPDH4 enzyme tetramer. Okay, so this is an example of good sticking or good quinary structure. Let's talk about an example where sticking could actually interfere with things assembling inside the cell. And the example I'm going to pick here is association of the SL2 RNA molecule with the U1A, that's a small protein RNA recognition motif, or RRM. And these form part of the U1 particle of the spliceosome. So the spliceosome is a quite complex organelle that assembles and disassembles every time to do the job of splicing pre-messenger RNA into messenger RNA. And the U1 particle, of which these two guys here are part of, the U1 particle is one of the first recognition elements that binds to the RNA. So there's an equilibrium between the bound and unbound state. And if you look inside, let me go forward here again. If you look inside mammalian cells, we have sort of a ionic strength or concentration of about 200 milligrams per milliliter. A large fraction of it is actually potassium ions. And indeed, one of the ways that you could try to sort of mimic in vitro somewhat more cell-like conditions is to take a reaction like this and look at its KD, the binding, the dissociation constant, as a function of potassium ion concentration. And so Caitlin Davis did that scan here. And not too surprisingly, you find that you get very strong binding at low concentrations of KCl. And then the KD increases. And when you're getting towards sort of physiological values, you have a KD that's a few times 10 to the -8. So increased salt decreases the binding affinity. This can be restored somewhat by crowding. So again, in vitro experiment here, if you look at, in this case, she looked at PEG 3550 as the crowding agent. And you add Crowder to this mix, including the potassium ions, you actually find that the KD decreases somewhat again towards around 10 to the -8. So there's a trade-off here between ionic screening and crowding. About a 10 to the -8 KD for this particular mutant of the protein and of the SL2 RNA that we were working with. So we can look at this inside cells. And so here's a somewhat fuzzy looking fluorescence image of a cell where you can see that the pair binds both in the nucleus as well as in the cytoplasm, although there's more signal from the nucleus. These are proteins that get imported in the nucleus, so that's not too surprising. And when Caitlin then measured the relative equilibrium, and compared the in vitro result with potassium ion and phycol, so this is kind of a good physiological buffer, versus what we actually see inside the cell, there's actually a more than 2 order of magnitude decrease in binding, so an increase in the dissociation constant, but a decrease in the binding that she sees inside cells compared to her in vitro experiment. And we think that what's going on there is there's, of course, thousands of other proteins and RNAs and lipids and so forth, as I said, inside the cell. And so not only do interactions have to evolve to bind, they also at the same time, of course, need to evolve not to bind to everything else, right? It's one of those things, if I have one thing that binds with an equilibrium constant of 1,000, but then I have 1,000 other things that bind with an equilibrium constant of 1, that's still going to be competitive from an entropy point of view because you have so many possible non-specific binding partners, right? And so we think actually there's basically here a case of non-specific binding or sticking going on that actually reduces the effective binding affinity between the RNA and the protein inside the cell. So this is one reason why maybe when one does, in some cases, in vitro measurements, you're getting these ridiculously great binding affinities, like KDs of 10 to the -10 or 10 to the -12 or things like that. They are probably not that low actually in the cell when these operate in the more complex environment with all the other things they can stick to. So let me go to my example number 4, preemptive chaperoning in cells by HSP70 and on time by HSC70. So the HSP70 family of chaperones, of which there are, I think, almost a dozen or so in human cells. There's only one in bacterial cells, a DNA K, DNA J, HSP70, HSP40 pair. But they consist of a ATPase domain and a protein bias. Of course, in our case, since we are looking at fluorescence inside cells, we've also hooked up an mCherry label to the chaperone, as well as a GFP label to the PGK. This is a known client of HSP70 that I'm going to use as an example. And we made sure that when we do in vitro assays by denaturant, sort of kind of standard assays that people do, luciferase assays, things like this, that they still bind. And it's not perfect. The binding is only about 70% of what you get with the unlabeled ones monitored by tryptophan fluorescence, but it's not a super strong perturbation of the system. You can certainly strongly perturb the HSP70 by making this mutation K71M here, for instance, that actually abrogates the ATP activity the ATPase domain, and therefore disturbs the ATPase mediated binding and release of clients to the chaperone. Okay, so we made a mutant that actually will melt around, or made a series of mutants that will melt in the high 30s to 40 degrees Celsius range, so kind of around where one would have a fever. And we looked at their activity in vitro and in cells. And so I'm going to show you both what happens with HSP70, which is a variant that gets upregulated on heat stress, and with HSC70, which is a variant that's actually latently expressed inside cells and does lots of other jobs. Like I mentioned, clathrin-mediated endocytosis, and it is actually involved in that, for example. It also interacts with the ribosome, nascent protein folding, many different kinds of things. So we have these two kinds of HSP70 type chaperone, one that's upregulated upon heat stress and one that's generally present inside the cell. And we looked at their ATP mediated binding to PGK. And what we found actually is there is no ATP mediated binding in vitro. So actually the in vitro conditions, even when we add HSP40 and ATP to the system, so there's a co-chaperone, there's plenty of ATP in there, we do not actually get proper reversible ATP mediated binding of our target to the chaperone. But when we do the experiment inside cell, we do. And so here are 4 different versions of PGK, a very high melting one in black and a lower melting one as the orange triangle. And we're monitoring the binding by FRET. And so basically what we're seeing is the high melting one begins to bind at a higher temperature. The low melting one begins to bind to HSP70 at a lower temperature. In fact, the temperature where the binding begins here is actually shifted about 5 degrees Celsius down from the unfolding temperature of the protein. So the midpoint for binding is about 5 degrees lower than the midpoint for unfolding. And so it looks like the chaperone actually binds the protein a little bit before it really begins to, a series fraction of the protein begins to unfold. When we do a control inside the cell with an inactive ATPase inactive mutant, we don't really see any significant FRET signal. And when we do the experiment with HSC70, so that's the latently expressed one, then again we see binding. But this time the binding curves actually shifted a little higher in temperature than the ones we see over for HSP70, pretty much exactly at the midpoint where the unfolding of the protein also occurs. And so our hypothesis in this case is that HSP70 is actually a preemptive holder for client proteins, whereas HSC70 has a later onset and really binds only once the protein significantly unfolds. And so we think that HSP70 might be actually recognizing these short hydrophobic near surface motifs that get even more exposed as the protein doesn't unfold yet, but dynamically fluctuates more as the temperature goes up. Whereas HSC70 really needs to see a significantly unfolded client to actually bind. And when you think about it, again, I can sort of hand-wavingly justify this in terms of protein evolution. HSC70 is latently expressed in the cell. It does a lot of other jobs. It would really not be good for its function if it just binds to a whole bunch of still folded proteins. And so it binds late. On the other hand, HSP70 is upregulated during heat stress. And so actually the quicker it binds to client proteins, even when they haven't completely unfolded yet, the better. So we think it's actually the fact that HSC70 has a lot of latent functions inside the cell, whereas HSP70 is upregulated, that makes HSP70 sort of bind even native protein pretty efficiently, whereas HSC70 needs a significant degree of unfolding. All right, example number 5. So I'm going to talk about cytoskeletal modulation of off-target proteins inside the cell. So there is, of course, a lot of proteins that are associated both with the actin and with the tubulin cytoskeleton inside the cell. But it could also have an effect on proteins that are not associated with the cytoskeleton. Again, like the PGK that I just mentioned in the previous example, that enzyme that makes ATP. And for the physical chemist like me, there's a number of drugs available that you can treat cells with that will either disrupt the actin or the tubulin-based cytoskeleton, so things like cytocalycin or lantronculin or nocodazole or vinblastin. And so the first thing that we did in our experiments is we copied a crowder from a very nice publication. Actually, I noticed I didn't cite it down here. I should put it on this slide. The only thing we did is we changed the crowder from having the original fluorescent reporters to having the ACGFP1 and cherry reporters on it. And the only reason we did that is because that's what we use on our other proteins in the experiment. And that way we don't need to change the color filters on our microscope setup all the time as we go back and forth between a crowding sensor and the protein. And so when the sensor becomes crowded, the helices sort of collapse together and you get a more fret between the two fluorescent labels. And so this is basically shown by this change in the donor over acceptor fluorescence ratio. So when D over A gets bigger, when I go into this positive regime, that means the green and the red move apart. There's more donor, more green fluorescence and less red fluorescence. So D over A gets larger. And indeed, that's what we see when we dissolve the cytoskeleton with drugs. On average, we actually see a change fraction of 0.4 in the D over A ratios, quite a significant shift over to less fret or to less crowding. This happens, by the way, despite the fact that when you actually measure cell volumes on a light sheet microscope of the same type of cell under the same conditions, that the cells actually shrink by 25%. So we have the cells shrinking, actually get less crowding. Okay? And that highlights is that the cytoskeleton, especially the microtubulin type cytoskeleton, plays an important role in also maintaining cell membrane shape and cell volume. And when you disrupt that, the cell shrinks and some water goes out of the cell, and that might actually intrinsically increase the crowding. But actually just having all the big crowders gone and having more fluidity in the cell from all of these smaller particles that are now floating around, apparently actually leads to a decrease, a net decrease in the crowding inside the cell. This also matches up with our measurements of two model proteins, VLSE FRET and PGK. PGK, again, is this metabolic enzyme that makes ATP that I mentioned before. VLSE FRET is not even a cytoplasmic protein. It's actually an extracellular bacterial protein from the organism that causes Lyme disease. And we sort of use it as a comparison, you know, like a cytoplasm protein, and they definitely not, you know, a cytoplasm protein. And what we found for PGK, in particular for the cases when blasting and nocardazole that affect the tubulin microskeleton, is that there is a decrease in stability. So we have a negative change in delta TM. The protein becomes a little bit less stable. And we've seen that before when PGK is less crowded. So less crowding, PGK becomes less stable. You add more thiol or you crowd it by some other way and PGK becomes more stable. So the result from PGK is actually consistent with what our crowding sensor is telling us, despite the fact that the dissolution of the cytoskeleton reduces the cell volume somewhat. On the other hand, VLSE doesn't really show much of a response here. Maybe, if anything, slightly in the opposite direction. It's within essentially almost the uncertainty of the experimental measurement. On the other hand, if we look at the D over A ratio, so how compact the protein is, there is a trend in the opposite direction. And so here our D over A ratio is actually positive. So again, more green means expansion. And so the unfolded state of PGK and actually even the folded state expands somewhat, worse for VLSE, if anything, a slight contraction. And when we look at folding kinetics inside the cell, dissolving the cytoskeleton has essentially no effect on PGK FRET, but it actually does slow down the VLSE by 50% or so by a time constant of 1 second when we do the experiment after dissolving the cytoskeleton. And so what we think is happening here is that PGK is more sensitive to crowding, but VLSE is also sticking to other components inside the cell. It's not a cytoplasmic protein, so it didn't evolve to not stick inside the cytoplasm. And this offsets the effect of the crowding. And then the sticking also slows down the folding kinetics somewhat. What we can't tell right now from our current generation of experiments, but we're thinking about it, and I'm sure Caitlin is thinking about it also up in Yale, is this due to a direct size reduction of crowders? You know, the fact that we, say, disassemble the microtubules into the individual tubulin monomers or dimers, or could it be due to an indirect effect that the disassembled cytoskeleton simply overall increases the fluidity of the interior of the cytoplasm and makes the crowding somewhat less. Maybe it's more crowding and less confinement might actually be one way that you could think about in terms of the mobility of the entities that are doing the crowding inside the cell. And that's what I'm showing this cartoon here, where there's a bit of microtubule shown, and then the dimers of the individual tubulin component proteins, and our PGK and our VLSE protein. So the upshot is when the cytoskeleton dissolves, cell volume can collapse, but crowding can actually decrease. And we can monitor this with proteins, and intracellular proteins don't seem to stick, but seem to be affected more by crowding. And extracellular proteins seem to be more affected by sticking, and it compensates for the reduced crowding. So as my last topic, number 6 here, before we reach the end for today, I'll talk a little bit about one more comparison, and that's folding in zebrafish tissues, which is an experiment that Rupei Feng and Caitlin did pretty recently, and we just published last year. And the idea here was to actually look at folding of the same protein as a model protein inside different fish tissues, like myocytes, muscle tissue, or keratinocytes, skin tissue, notochord tissue, islands tissue, and these different tissues have different amounts of crowding. For example, the islands is one of the most crowded tissues in an animal, whereas myocytes and keratinocytes are sort of much more typical of the kind of crowding you get in somatic cells in animals. And the way that Feng and Caitlin did the experiment is we basically want to make a fish that expresses the protein whose folding we want to monitor, but preferably just in a handful of cells, even just one single cell, like the one that the arrow points to in this picture. There's a little bit of autofluorescence from protein in the gut also, but I'm interested in this cell over here. And the way they did this is by making a plasmid that contains a tissue-specific promoter between two ICI cutting sites. ICI is a meganuclease that has a very long recognition sequence that doesn't occur in higher organisms. And so, and then there is a SV40 polyadenylation sequence just to make sure that the RNA can mature. And so basically what happens, this can get inserted into the tissues of the embryo when they inject the embryo, but not very efficiently. It's not very likely, for instance, that there's actually any germline transmission, and we don't breed the animals after they've been injected, so we don't even call them transgenic. But what it does do is it will go into a small number of cells, and the protein of choice, in our case again, FredPGK, is only going to be expressed in those cells for which you have put a promoter into your plasmid. So in this case, I think it was actually a muscle tissue, and they've done it with other tissue types. And so we can put the fish under the microscope and actually do a temperature jump experiment on the animal. And we can do this at temperatures that are in the 20 to 40 degrees Celsius range by using proteins that are destabilized, like this mutant of PGK that we use here melts probably around 37 degrees or something on that order. And so we can actually measure both kinetics as a function of time and actually protein stabilities inside these different tissues. And we can do it on a cell-by-cell basis and even look at cell-to-cell variations inside the tissue. You can see a few cells here that happen to have been transfected and are expressing the protein. And you can see the nucleus here in the cytoplasm of each cell. And so when we compare myocytes and keratinocytes and notochord and islands cells, and here are the actual cells from their experiments, we find that there's a range of about 7 degrees Celsius in the stability of a protein when you go from the in vitro conditions through cancer cells to myocytes, and then eventually end up with islands cells as the ones where the protein is the most stable. Okay? So a significant range of stability. And again, it isn't too surprising to us that island cells ended up in the most stable, the yellow ones over here. The reason being they have a crowding that actually gets up into the 400 to 500 milligram per milliliter range, as opposed to the 200 to 300 milligrams per milliliter that's more typical for most cells. So these island cells are really a very high crowded environment for this protein. And as we saw, PGK is crowding sensitive. It tends to become more stable upon crowding or less stable upon uncrowding, as we saw in the previous example. Here it's becoming more stable upon crowding in the islands tissue. Now for PGK, this probably doesn't make much of a difference because the natural PGK has a much higher melting temperature anyway. But imagine things like disordered proteins or things like that, or protein-protein interactions that are temperature sensitive, right? In some organism like that has homeostasis, like a mammal, let's say. And you can see even in this little diagram here for PGK, there are certainly temperatures here, like 37 degrees Celsius, where in one type of tissue, the protein is totally folded, and in another type of tissue, it's half unfolded in the myocytes. And so one can actually imagine that things like osmotic pressure, temperature, crowding differences between different tissue types could actually lead to different degrees of protein compactness, folding state, or binding between RNAs or proteins, depending on what kind of a cell they are in. So this leads to an even richer variety of possible functions for proteins, because you don't even have to do a post-translation modification or something like that. Just having the protein be in a different cell type can actually change its compactness or its stability and maybe its function. We certainly didn't do any experiments here that actually look at protein function inside these cells. Okay, so it is now 10:52 on my clock, and that means it's time to wrap up. So I'm going to stop here and just leave you with this summary as a reminder of the various things that I talked about in your slides, in case you want to ask questions about one or the other topic. You have like a sentence in front of you here talking about it. So with that, I'm done. I thank again all the people in my group, the NSF and the NIH, for supporting our research, and I'd be happy to answer any questions you might have. Ben, it's yours.
Thank you very much, Martin, for this beautiful overview. And I'm sure you can hear the digital applause in the background. And with that, we have time for questions. And so what I will do is, well, I have, if you have questions, please just mention this in the chat, and I will just go through the questions that are in the chat in the order that they arrived in, and then ask you to just unmute yourself and ask the question. So the first question comes from David Nesbitt. David.
Yeah, Martin, fantastic. Just, so full of physical insight. I want to go back to the very first example that you gave, where you had really this power law looking plot for the stickiness of your protein folding. The question I wanted to ask was, is there a physical understanding of what that slope means for your protein? And is it really, you're only looking over maybe 2.5 orders of magnitude in the time domain. Is that enough?
Yeah, exactly. So, I mean, that's a good point. I mean, it does do a pretty good fit. Can everybody, by the way, see the slide from the talk? I'm still screen sharing, just want to make sure. First of all, the caveat here is we're looking at 2.5 orders of magnitude only. And to distinguish power laws from stretched exponentials or even exponential, you need a fairly large time range to be able to do so. This one fits pretty well to this black dotted straight curve of an exponent of -1. And of course, we were wondering, like, hey, why -1? Right? 0.3 or 0.8 or God knows what any. We actually found an interesting paper that we reference in our paper here that actually shows that if you have objects that can make a contact with one another via a random size patch, so that the interaction potential between them randomly varies every time they make contact, which is something you might expect if you just have things that aren't engineered to contact one another, contact each other in various places, that the resulting distribution in interaction potentials is guaranteed to produce a t to the -1 power law. And so actually we looked at this and went, hey, somebody already figured out the explanation for this. At least this makes perfect sense in terms of what we think we're seeing here. So actually the very simple ingredient of having round objects with variables, randomly variable size interaction strengths will actually produce this kind of power law that has exactly a coefficient of -1. And that was one of the motivations why we figured.
Good, thank you.
So,
I will email you another question, but I'll let other people ask questions now. Thank you.
Okay, so the next question is from Moha is the short name. So Moha, do you want to unmute yourself and ask your question directly?
Nancy? Hey,
No?
Nancy.
If not, then I will just read the question, but I'll wait for Martin to return.
All right, I'm back. Well, we'll
Okay.
try number 3.
So there's a question here that I'm reading to you, Martin. A question about stickiness. Did you observe the limit at which interaction between different partners led to adverse effects for one or both partners? For example, protein is denatured. Limit as an interaction time or interaction frequency. My mic does not work, so please ask the question for me. Okay, so now I read this. Good.
Ah, perfect. So I'll go back to this slide here to try and answer that. So I think to be honest with you, the short but truthful answer is we don't really know yet. And the reason is that even with like 200 microseconds of Anton 2 simulation, we did not observe enough of these events to really securely establish a correlation between like the frequency of these events and you know, how likely just surface binding versus actually, you know, remember I talked about this effect where actually you can form new secondary structure that's non-native between 2 different proteins as an example in the sticking. So, you know, we're at a stage where we have observed these examples in simulations, but there's a lot of work to be done to actually quantify that. You know, how common is this? What types of proteins? Are there like surface markers, you know, electrostatic or otherwise, that might indicate the propensity for these kinds of things happening?
Okay, the next question is from JM Delfino. Would you like to ask the question yourself, or shall I read the question? So if you want to ask the question yourself, maybe just unmute yourself. No, then I'll read the question. Is the 14 micromolar GAPDH PGK affinity consistent with finding these 2 enzymes as a permanent complex at cytoplasmic concentrations by comparison with other glycolytic enzymes known to be isolated and not complexed?
Yeah, so let me go to that slide here again. So yeah, PGK and GAPDH are among the most abundant proteins, both in prokaryotic and in eukaryotic cells, with on the order of 10 micromolar concentrations inside the cell. I don't remember now the exact number, but it is actually, it's been studied at least in several different types of eukaryotic and prokaryotic cells. So the number that we found, and this is an effective KD that I've reduced to a micromolar units again, so it corresponds to a 1/1 value. We actually do see 2-to-1 binding, right? So it really, there's a KD with micromolar squared units, but I sort of reduced it by concentration here. But basically, yes, the in-cell concentration is actually on the same order as the KD here. And so we would expect pretty significant and frequent transient association between these 2 proteins under the real conditions in the cell. And this is a good question because, of course, the proteins we did the experiment with here are not the natural in-cell proteins, but they're fluorescent ones that we've stuck in there. We can actually do these experiments also when we do temperature jumps or other things as a function of concentration inside the cell, simply by taking cells with dim green and red fluorescence and cells with high intensity dim, green and red fluorescence, and actually even do concentration scans that way. But in this particular case, yes, there's a significant degree of association at the actual in-cell concentrations of the real proteins that would normally be expressed in cell.
Thank you, Martin. The next question is from Gonzalo Pratt-Gay. Would you like to unmute yourself and ask your question directly?
Yes, hi, excellent work, in particular the quantitative aspects of folding parameters. And related to this, given the crowded environment involved, can you tell when the TM corresponds to unfolding and when it corresponds to denaturation by aggregation?
Yes, and so I don't have a slide here for that. And you know, we actually usually cut that out. I'll be totally honest with you. We often cut that out of our little plot when we show the unfold curve. So the way these curves look,
Martin,
is
sorry.
some baseline, but
Martin, you were cut out for the last
sentence.
Can you just repeat the last one?
I will repeat again. Can you hear me again? Okay, so these curves typically start with some baseline in the folded state, and then it goes up at a certain temperature, and it becomes flat again at higher temperature. And so what we actually see, though, if we go to even higher temperature, and this goes to your point, Gonzalo, is that this curve will actually then come down again. So you'll actually get more fret again. And that is because finally at sufficient high temperatures, the denatured proteins will actually begin to aggregate. So initially you see an increase in the green fluorescence as the proteins dissociate and there's no more fret. And then you see actually the red fluorescence come back. And in some cases, we actually observe that the fluorescence goes to a nice baseline at high temperature. And in other cases, we actually observe when we compare it to in vitro measurements that it actually already begins to turn around when it's still halfway through its unfolding curve. So there are some proteins that don't aggregate as quickly inside the cell where you can kind of see the full curve. And there are other cases where actually the aggregation begins almost as the unfolding begins and the curve sort of turns around early. And so we can see a signature of that, yes.
Thanks.
Our next question is from John Muller. Would you like to unmute yourself and ask the question?
Oh, yes, yeah. So I really love this quantitative approach for GAPDH PGK interaction. But as a lecturer in biochemistry, what consequence does it have now on really the reaction? So is there substrate channeling in there or not?
Yeah, so from our experiments, we honestly do not know, right? All we did is measure this association. And so it remains to actually be seen whether this association constant does lead to substrate channeling. There has to be still a functional assay done. It's plausible because the concentration of these enzymes is on the order of the KD that we measure for them, of the real ones, not of ours, but of the actual expression of PGK and GAPDH in the cell. But we have not done a functional assay yet. It is possible to do, though, so that's the good news. There is actually a fluorescent NADH analog-based assay for this reaction that one can actually use to look at the reverse reaction of the, and so that might actually be able, one might actually be able to use that even in cell, but we haven't tried. So, we can only say it's plausible. The functional part remains yet to be looked at.
And then here we have a question from Kendrick Nguyen. Kendrick, would you like to ask the question yourself?
Hi, Professor. Very, very interesting talk. Thanks a lot for the talk. I have my question is kind of two parts. The first part is it better to describe these interactions as being modulated by salt concentration or by ionic strength? I know that there's a differentiation between the two terms, but is there a preference over which one should be used over the other? And the second question is, can you please comment on whether and how ionic strength or salt concentration can modulate the so-called quinary interactions between light charges? Thank you.
Yes. So what we would expect, I'll start with part 2 here and go where was it? To that slide here. So here we have this like charge effect. And so you would actually expect that screening will simply increase at higher ionic strength. And this is going to probably be irrespective of the type of ions, whether it's, let's say, sodium ions or potassium ions doing it. And so with more screening, you're going to get a potential that looks more like a Yukawa potential or Debye-Hückel type potential, or e to the minus AR over R instead of a Coulomb potential, of course. And that's probably also going to affect the screening length at which you will finally drop down into this minimum. Now, as far as the question of just ionic strength versus salt concentration, we have actually done some experiments where we can temporarily pump sodium plus versus potassium ions, potassium plus inside cells. Because of the difference in the number of sodium and potassium pumps, you get different results as far as, cells can very quickly recover from one and not from the other. So it's very easy to apply osmotic pressure with, let's say, sodium, but not so easy to do it with potassium. Same thing with sugar versus mannitol, for example. And experiments not really seen a large difference. So I think, at least for the measurements that we have done, just talking about overall ionic strength would be good enough to describe the situation. However, I mean, we all know, right, there is a very large imbalance between potassium and sodium concentration inside cells versus the extracellular milieu, you know, a lot more potassium ions inside the cell. And I'm sure it has its good reasons, as all biochemists know. And so my guess is that with some more careful looking at it, people will actually find differences between merely looking at overall ionic strength versus actually looking at what types of ions are involved. But we haven't seen it in our experiments.
Thank you.
Our next question is from Tanumoy Mondol, and who asks me to read the question. When you measured Fred efficiency change of HSP70 PGK binding with increase in temperature, do you think increasing temperature, this heat shock, induces conformational change in HSP70 itself? Does HSP70 sense this heat shock or increase in temperature compared to any temperature-dependent general protein conformational change?
So the answer is yes, HSP70 definitely also senses the temperature change itself, and it depends on the ATP concentration. So if you do the experiment at very low ATP concentration, there are actually structural changes that look like a aggregation or collapse of the protein that occurs low 40s, 40 degrees Celsius. Under the kinds of ATP conditions that occur inside more healthy cells, millimolar concentrations, the transition temperature for HSP70 goes up above 50 degrees. I don't remember, somewhere between 50 and 55 degrees Celsius. Now, in the experiments that we did, the ATP concentration was high enough inside the cells, and the same was true for the in vitro measurements that we did, that the HSP70 wasn't near that transition. So these transitions that you're looking at here that start like in the low 30s, HSP70 will at the earliest show a transition around here at low ATP, and a transition actually at 50 degrees outside the range of these measurements, where the cells will die right above 45 degrees or so. So the answer is yes, there is sensitivity. There is even some sensitivity in the fever range, and this sensitivity gets higher when the ATP concentration is lower. But that was not the case for our experiments here.
Thank you very much, Martin. The next question is from Gilad.
Yes, Martin, thank you for this exciting talk. And I have a question which might be a bit technical. So over the years, you've done many beautiful experiments with these fluorescent proteins as donor and acceptor for FRET. When we do FRET, we deal a lot with things like the kappa squared and the spectral overlap between the donor and acceptor and other factors like that. And I was wondering how these might affect what you measure and whether you have a way to even look at these effects inside the cells, inside these crowded environments.
Yeah, so you will notice, right, that I have carefully stayed away from using the FRET measurements in our experiments as rulers, for example, because anisotropy of the two proteins with respect to one another is going to have effect on doing distance determination. In some of the experiments, by the way, if you look here, I'll see if I can find the HSP again. You'll notice that our FRET only changes by 8%, right, 1 to 1.08. Of course, it would be much nicer if we could do the experiment in a way that we can get like a 40% change or something like that, right? The reason we settle for that often in our experiments is it's even more important for us than a large FRET signal change or a large FRET signal is, of course, that we don't want to mess with the proteins, right? So if I go to the previous slide, when we hook up our labels, we want to make sure there is still chaperoning action and so forth, right? You can't label these proteins just anywhere you want. So there's always this problem of, if we could label it in the perfect spot, then the thing doesn't seem to function very well. And so we have to settle for spots that may not give us the highest change in the FRET. So very often in our experiments, like with a FRET change of 8%, you certainly would not want to do distance measurements or worry about things like this. This really becomes more of an on-off thing. That said, we do not usually actually try to do polarization measurements. It would actually be a very useful thing to do because there is additional information to be gained there. So inside the cell, even a set of labels that has a more or less isotropic kappa value, whatever 1/3, whatever 2/3, whatever it is, that can sort of tumble around, when they actually get stuck to other large, slow-moving things, that could actually increase the anisotropy. So we thought about doing experiments like that to extract some more information out. But to be honest with you, we have settled for this kind of very averaged information so far.
Okay, thank you.
Now, our next question is from Samantha Nernay. Would you like to ask the question directly?
Can you hear? Am I audible?
Yes, you're
Yes.
audible.
Okay, so first of all, thank you very much for this, Professor, for this great talk. So actually, I'm wondering how that's, you told that cell shrinking can decrease the cell crowding. So can you please explain? Maybe I missed, I think you explained a bit, but can you?
Yeah, so are you talking about the cytoskeleton?
Yeah, so
example?
you told that, during the frame measurement, so cell shrinking can decrease the cell crowding.
Okay. So here's the thing, right? There are sort of two competing effects. On the one hand, when the cytoskeleton dissolves, you're making a lot more of those monomers, right? And those monomers could actually crowd. Not only that, because the cytoskeleton dissolves, in our case, we actually observe cell shrinkage. And so actually cell volumes reduced. That again is actually something that could in principle increase crowding. So there are certainly things going on like producing more smaller monomers and reducing the cell volume that could have actually increased the crowding. Nonetheless, we found that our crowding sensor actually indicated reduced crowding inside the cell. And so what we think is happening here, among other things, is that the intact microtubules and actin filaments also inside the cell rigidify the cytoplasmic environment to actually enhance crowding. Whereas if you have a larger number but of small molecules that are doing the crowding, just the monomers or the dimers, you have more crowders available, but they're not as effective as crowding as when the cytoplasm is sort of rigidified. And so we think that that effect simply wins out over just merely counting the number of crowders. And there's two proteins that say that that's the case. There's both this crowding sensor, and we've done a lot of other experiments with PGK, right? The one here that becomes less stable, where we've shown that every time you crowd it, it becomes more stable. When you uncrowd it, it becomes less stable. So actually our crowding sensor and the PGK both make that same indication that dissolution of the cytoskeleton actually makes for an effectively less crowded cell environment. And we think it's simply because this indirect effect here of disassembling the cytoskeleton affecting the fluidity of the cytoplasm may be the reason for that.
And do you think that we can consider that during measurement? Because we do really observe that cell shrinking, but do you think that is there any way to consider that or some kind of control experiment? Or we cannot?
Well, yeah, so in a sense, this is actually sort of a control experiment for that, right? So latrunculin and cytocalysin actually have more an effect on the actin cytoskeleton and vinblastin and more on tubulin. And actually you can see the effect is smaller, possibly even non-existent in that case. And again, tubulin is much more massive, microtubules are much larger than the actin cytoskeleton. So again, we think it's actually the rigidification from the very large cytoskeletal units of the cytoplasm that might be the reason why crowding is more effective when the cytoskeleton is intact than when it's not intact, even though dissolving the cytoskeleton actually produces its own extra little crowders. So.
Okay. Thank you very much.
Okay, so we're
the paper's coming out, by the way, in a few days, I think. There's a lot more detail in there.
So we are approaching the end of our list of questions here. The next question is from Hagen Hofmann.
Well, Martin, thanks a lot. It was really a wonderful talk. And my question is very much related to the previous one. In the beginning of your talk, you mentioned that entropic crowding effects of a few KT are sort of in the same order of magnitude as the interactions that you find for quinary interactions. Now, after having seen your talk and having seen so many examples where quinary interactions play an important role, and also one example that sort of contradicts what you would basically predict from crowding, namely disassembly of microtubules, where it's not really clear why you actually get this effect despite the cell shrinking. So would you say that quinary interactions actually are more important than crowding, or would you still keep the balance between the two?
You know, in the case of the proteins that I talked about here, clearly, for instance, for VLSE, quinary interaction or sticking is more important, right? Because we don't even see any evidence of the uncrowding effect that we get with PGK. And we see evidence of sticking because its folding time actually decreases significantly. We don't see it for PGK. So I would say in this example here, though, still crowding is probably as important or maybe even more important than sticking. And in the case of VLSE, it's the other way around. So I think here's a case where, you know, you're seeing sort of both extremes. So, you know, these are only two proteins, right? And really the number of things that we and others have looked at in cell, the number of proteins, the number of different assemblies is still pretty small. So I would say the word is still out in the end on how important, you know, both of these are going to be and how they're going to balance. So I'll, you know, yes, it could turn out that sticking in the end is going to be more important and more prevalent. There's lots of different ways that you can stick. I think the verdict is still out.
Thanks, Martin.
So Martin, if you're not completely exhausted, there would be two more questions. Another one from Moha, a question about the cytoplasm environment, if there is time. How do the components of the cytoplasm mimic interact with and affect one another when there is no extra protein present? Is there any discernible change in the structure or arrangement of the components during the long simulation? Would you keep the components restrained during the simulation?
Okay, so I'll make the long answer short. We published two papers. One that came out this year looks more at the folding question I was talking about, the WW domain interacting with various things. The first one here, JPC Letters 2019, actually discusses a little bit more what comes up in that question. And so what we did in that paper is actually we analyzed a large number of cytoplasmic proteins and how they stick and interact with one another. And this is actually where that particular plot comes from. So that's not just for WW domain. This is really for all the pairs of proteins that can stick in the cell. One of the things that we noted in our simulation, which is also an indication of a little bit of excess sticking probably, is that the proteins tend to cluster. So we have about, I think in that simulation, somewhere around 300 milligrams per milliliter of occupied volume. But when you actually look at the full simulation box, and the data is in that paper, so you can actually look at, the simulations are also available. If people want to analyze the simulations themselves, you know, the information will be available on the web. What we actually saw is that the proteins kind of sort of clumped up a little bit in the box. So there were areas that had more water in them and areas that had more protein in them. And I would expect the real cytoplasm not to be quite as clumpy, but maybe a little bit more homogeneous in the sense there aren't going to be like big water pockets or anyways large as what we see in the simulation in it. So that might be just an artifact of the force field. But the answer is yes, we can see these kinds of things in our force field, and you know, take it with a grain of salt of the current generation of force fields maybe being somewhat too sticky when you're seeing things like extensive protein clustering.
And then if I may, I have a final question. I was very impressed by your result about SL2 and U1A, where you saw 100 times weaker binding in the cell than in vitro. And so you also dissected it into on rates and off rates. And so I can still imagine that, for instance, the off rate is a quantity that can be measured quite reliably. For the on rate, it's a little harder, I imagine, because the observed on rate will depend both on the concentration and the actual rate coefficient. And so my question is essentially, is there a contribution from just stuff sticking somewhere that effectively reduces the concentration of the binding partner, and that leads to a lower observed on rate?
Yeah, so that's actually what we think is happening. And you can see our estimate here. That number, by the way, you know, Caitlin is still working on the data analysis. We'll see if that's going to be exactly the number that we'll put forth. But I think what you just said actually in a nutshell describes how we're thinking about this right now. Namely that non-specific sticking interactions in the cell, and so again, Hagen, yes, sticking here, non-specific sticking interactions are effectively you know, sequestering either the RNA or the protein, and therefore preventing the on reaction or slowing down the on reaction. And that's a major contribution to that change in the KD. Yes.
And I mean, it's sort of almost a philosophical question, but is it then still fair to say that really the KD has changed, whereas you're essentially just competing with the KD to other things?
Yeah, but this is basically how you can think of it. I mean, the KD that we measure here is basically just the effective KD, right, that comes out when you just pretend the two things are just on their own, right? So the increase in the KD comes from the environment. It's due to the effective non-specific interactions that prevent things from coming together. Exactly. So if you want to think of it that way, the KD of these two, of the RNA and the protein, the KD is the KD, right? And it's only the environment that makes the apparent value of the KD actually higher.
Yeah, well, thank you very much, Martin. And I think there are no further questions. I hope I didn't overlook anyone. So with that, thank you very much again, Martin, for this very impressive and beautiful talk. And thank you all for joining in and for the lively discussion. And I would like to invite you all back in 2 weeks for our next talk, when we hear about NMR, protein folding intermediates and the like from Jane Dyson. And with that, I hope you all safe and stay safe and all the best, and hope to see you again soon. Goodbye.
All right, I look forward to it. And Ben, Gilad, and Hagen, thanks for organizing this. Bye, everyone.